Thank you so much. I want to thank the organizers very much for giving me a chance to speak to you all today, and thank you all for your attention as we round out this session of this amazing conference. I am really excited to have a chance to talk to you today, not about anything really getting specific into the weeds on any specific workflow or architecture or bit of code examples, but instead I want to zoom out, and I want to tell you a little bit about our experience about running these workflows at our institution. And the goal of my talk is to really just give you some perspective, because at the end of this conference, you're going to go back to your institution, to your company, and you're going to be wanting to run these workflows that you love in a way that gets them into the hands of the right people, that gets the data in the right places. And so that means that there's a much larger context, and that's what I want to share with you today. So the context that I'm working in is the Fred Hutchinson Cancer Center. We are a nonprofit research institute based in Seattle, Washington. We have a large number of basic research labs, over 200. We have a large number of clinical providers. There's a whole hospital that's a part of us. We also have a great centralized core facility system running genome sequencers, mass spectrometers, microscopes, pushing data out to drive all these research activities. And it's really a massive undertaking that I've been happy to be a part of since 2017. Here's a great little photo. This is a beautiful sunset photo that you get around maybe like 3 p.m. this time of year. But it's a wonderful part of the world, and I've really fallen in love with it. And the group that I'm in is trying to address this challenge, which is not unique to us. This is the challenge of the moment for people sort of like us, which is how do we remove any barriers to research and development and progress which are being posed by the scale, the scope, the complexity of working with this amazing data that we're able to generate. I think the talk earlier about the scale of genomics and what's going to be required to process 100 million personal genomes was really a fantastic example of that earlier today. Some of the key highlights are the cloud is amazing, but it is complex, as we just saw. The audience of the people, the different systems that need to use these resources are quite diverse. And also, there are real security and cost concerns that need to be addressed. These are extremely, extremely important to making the whole system work together well. In my specific institution, I like to think about what is the audience of people that I need to be able to satisfy the concerns of in order to be successful. And one group that we're probably all very familiar with would be something more like the bench researcher, someone who's working in a lab. Their goal is to publish papers and get results. They're not super interested in learning everything on the command line. They need things to be straightforward. They want to be able to process their data with robust tools, be able to share effectively and move on with their life. We are also really concerned with the success of our core facilities, the people running genome sequencers, running these microscopes. They want to be able to generate the data, deliver it to users, let the users get the most out of that data, and then be able to, without disrupting the processes that they've already built up over years and years and years for making these amazing facilities run well. The third group is one that I personally identify with most. I won't ask you to raise your hands, but I suspect a number of people here would think about as being the bioinformatics staff. These are a very important part of this ecosystem. The people that are actually writing the workflows, the people that are being tasked with figuring out how to bring the data together. Anytime someone says they're doing multimodal analysis, I think I'm so happy for you. That sounds like a giant pain. And the people that are really tasked with making sure that we're getting the discoveries out of the analysis. And some of the challenges that I've had in that role over the years have been, how do I do something amazing and then put it in the hands of the bench scientist? How do I do something amazing and give it to my PI? How do I get data from Genomics Core effectively? And also, how do I work with my IT infrastructure? And I think that's the last group that really deserves to be mentioned. So when I think about our IT, we have an amazing centralized IT system at Fred Hutch. And I think these are people that it's easy to forget about because when they do their job well, you're not talking to them. When the trains are running on time, you're not thinking about it. And when you want to build something new, you end up having to talk to them and say, well, yes, I want this to be secure. Yes, I want to make sure that people can log in with the right credentials. Yes, I want to make sure that the right cost controls are in place. Being able to satisfy their requirements is honestly, it's a big challenge, and it's something that deserves a lot of consideration when you're thinking about the overall architecture of how workflows and analysis are running. So these are a lot of challenges, there's a lot of people. The group that I'm a part of, just by way of introduction, is actually relatively small. There's a much larger number of people at the larger organization that are a part of this. You know, we're obviously very collaborative, and we've had a lot of great contributors. But the core team are shown here. We all come from different backgrounds. So I'm a basic scientist. I'm a microbiome. I'm a sort of metagenomics person by background. We have we have someone with expertise in cloud engineering, web development. We have an astrophysicist on the team. We have someone who's done industrial design. We all come at this from very different perspectives, which means that we all have really different ideas about how we can solve things, which has been, I think, part of the reason we've been able to come up with the ideas and the approach that we have. So when we considered the landscape we were dropped in, we considered the goal that our organization had given to our group specifically, and what we ended up deciding to move forward with was building a data platform, which is called Cirro. So I'm going to talk about this. This is Cirro. This is something that it's a serverless application. It's running inside our AWS. It's all AWS based, and it's all internal within Fred Hutch. But the idea is we want to have a place. We want to have a utility that can address the concerns of all the different groups that are involved in generating, analyzing, distributing, sharing, and managing these really complex data sets. When we started out this process, we wanted to keep some really core principles in mind. So the first is we want to build as little as possible. So taking advantage of managed services offered, in this case by AWS, because that was the provider we decided to go with, that was something that was really great for us because we were able to, for example, say, we're going to use HIPAA-eligible services for all the individual components. That way, we're pushing off the responsibility for managing the security of those services to someone else, and that's not in our domain of concern. It's nice to think about what you can spend time not building, right? We also wanted to keep things as simple as possible. Every time someone would build something up in the display, someone else would come down and try to rip out everything that they didn't think was necessary. We've gone back and forth. It's constantly in tension. I'm usually on the sort of the add it side of things, and other people on the team are on the rip it out side of things. So sometimes there's feelings involved. But simplicity is something that I think is really, really important for communication so that people can understand what they're looking at. A big part of this process has been listening to users. And for me, this means sitting down with scientists, not just building something and thinking, oh, it works, but instead saying, let's see what happens when I sit down next to the person who's supposed to be using it, and I watch them open up their browser, and I see where they click, and I see what they think should be happening. And to me, this is really educational. I spent my whole career in bioinformatics in the command line, and I think a bunch of things are obvious, and they're not obvious. They're like really hard to figure out sometimes, and that's purely about the attention span we all have. We all have a limited amount of time to understand things, and every additional thing you ask someone to understand is taking away from their attention budget. So you want to be able to make as little ask as you possibly can. Another big part of this for us has been collaborating on science. One of the big drivers of our effort has been picking specific scientific projects that needed a boost to be able to go further, go faster, and to be able to collaborate with those people to figure out what can we do to make this project really impactful, with the hope that what we build will then be able to provide benefits to others. So being embedded scientific collaborators is not only personally satisfying as a sort of ex-scientist myself, but also a major driver of our design decisions. So I'm gonna walk through features, and what I want to do is just 6 of them here, and I'm going to point out sort of the people that they are intended for and really why we implemented them in the context of our specific organization. And I will say, everything that I'm going to show you is very much thought of as being for a biomedical research institution. There's many different types of organizations, and I can imagine that this would look very, very different at different organizations. So the first one is data ingestion. There are a number of different places that data can come from, and we wanted to have a single place where they can land coming from different routes. So a very important one is dragging and dropping from the browser. You want a user to be able to say, hey, this data set's only 10 megabytes, why can't I just move it over? let's make sure that can happen. People often want to be able to pull data from public repositories, so thank you NF-Core Fetch NGS, big fan. Pull data from public repositories, very happy about that. People want to be able to upload from the command line. A bunch of stuff can't go through the browser, so having a command line interface is obviously essential. Something that I didn't expect is actually having hardware integration, and I'm gonna mention that in a bit in another slide. But the idea that we can have a hardware device which is also able to automatically push things up into the platform. This is all built on AWS Batch and AWS S3, so object storage. The next, so getting data in, this is sort of something you can imagine is very simple. Actually, one sort of nuance here, because I don't think I'm going to run out of time. One of the design decisions we made really early on that I thought that maybe would be appreciated by this group a little bit more than other people would be the notion of how we organize data. And we decided to say we're going to think of data as being organized into data sets. Okay, sure. A data set is a collection of files. That's it. A data set is a collection of files. We're entirely file object oriented. Maybe one of those files can be a Parquet file and you can treat it like a database. That's fine. But then when we have a collection of files, we get to do very, very minimal typing on it. And it's entirely sort of like file name structure. So if I say I have a paired end FASTQ, sorry, if I have a paired DNA-seq data set, it's going to expect files that are in FASTQ format. That's the format. But also, that data set now is eligible to be analyzed by workflows which are expecting paired DNA-seq data. So it's, so we have, I don't remember exactly how many different data types we have here, but we have things like for images, I think up on the screen it says RNA-seq right there. So FASTQs could be DNA, could be RNA. Why would I call those something differently? Well, it's because I don't want to upload RNA-seq data and then ask, oh, am I going to run serif variant calling on it? It shouldn't be run on it, right? It's the wrong data type. And so we want to pair the workflows with the data type. Okay, that was a bit more of a digression than I anticipated. But getting the data in is actually a little bit more interesting than I thought it would be. Big part of this, scientific workflows. We're all here to talk about workflows. When you want to, I spent a long time trying to help other scientists analyze their data. When I got to the Hutch, I was a very early adopter of Nextflow. A lot of people call me, oh, that guy who does Nextflow. And a lot of times I was saying, hey, you want to do a thing? I'm not going to do it for you. I'm going to write some Nextflow. I'm going to give it to you. I'm going to point you to NF-Core, and I'm going to train you so you can run it on the cluster. That worked sometimes. It worked much better than anything else I'd ever done before. The most success I had ever had with helping other people run workflows, I think by far and away NF-Core has been the most successful thing I've ever had. And I think, honestly, the reason that I was given the opportunity to do more in my organization was because of how useful Nextflow has been for researchers. And I think that when you think about the collection of researchers who need their data analyzed, you'll understand that there is a segment of people that will run things from the command line, and there is another segment of people who haven't learned the command line, and asking them to log onto the cluster is asking them to learn something new. And so when I say that it's worked for some people, I'm not being trying to be facetious. What I'm really saying is there is a broad collection of people who want to analyze data but don't know the command line. And I think that there might be some debate as to whether or not you put analysis tools in their hands or you ask them to collaborate with a bioinformatician. I'm agnostic on that. But we did want to make a point-and-click interface for being able to launch workflows. So a lot of this is, obviously, a lot of the foundation of this has already been pioneered in NF-Core. You look at the schema for parameters, and you say this is actually very, very close in format. Unfortunately, not identical, but close in format to the React JSON schema that you would use if you want to generate a web form, right? And the NF-Core schemas are used to generate web forms, which is great. So we use that as a starting place. We had to add a number of different sort of like platform-specific things. We want to say, let's make a place where people can click on a data set, click on a workflow, select all the parameters, click run, and have that just launch inside their account. That's very important. Another big part about running workflows has been letting people add their own workflows. So giving them the tool to make their own form, specify their own workflow, click add. And this is, I think, the tool for people like us, for bioinformaticians. I want to make a new tool, and I want to give it to my collaborator and say, you run it. I'm not running it for you. You run it on your data. I turned it into a button in the data platform for you. And that's something that I find really satisfying when I see bioinformaticians at my organization who have started doing that. That's very early days. Okay, so that was the workflow digression for this particular conference. Big part of the platform has been connecting directly to instruments. So we have a hardware device on premise. It's a storage gateway from AWS. You drop files on it. It just looks like a shared folder. Those get streamed up into AWS. You can set up a we have set up a Lambda trigger in there, which puts them into the right account. What this means is data comes off the instrument. So I think that flow cytometers were the first ones we did. There's a number of other ones that are going through, like proteomics data. They drag and drop onto a folder. It goes into the customer's account, the lab's account, and then they get an automatic email saying, hey, your data is available. Click on this link to go find it. That's really huge for automating and streamlining processes from the core resources, which I think are a huge asset at Fred Hutch. Being able to say workflows are great, but there might be other types of compute you want to run. So you might want to launch an interactive notebook. You can do that in Cirro itself. We have a SageMaker interface, which is nice. Being able to pick what kind of compute instance. I want to do something big. I'm going to rent a ½ terabyte of RAM, but I'm only going to do it for a couple hours. That's wonderful. Also being able to pull the data down directly in Python or R and saying, I want to run my custom stats. The data table I'm loading is actually a file in my account in Cirro. I can access that in an authenticated way and just load it directly into my live Python session. I think is a really nice way of making sure that the canonical copy of the raw data always is in this one place. You can control access to it. And then I know, hey, I ran this analysis. If I give this notebook to my buddy who has the same access, they can run it. And I know that the input data is coming from the same place without copying files around, which is nice. Getting to the larger organization. So that's something this is much more intended for sort of the bioinformatics people that want to get into an interactive session. Account management turns out to be huge. So this is really, how do you give people access to the data that you're delivering to them from the point of view of the bioinformatician? And how do you, as a PI, make sure that your lab members have access to the right data? How do the IT professionals ensure that each lab is only seeing their data with proper security? And also, how does IT get out of the business of answering tickets to add and remove people? A PI says, I want to add person X. IT doesn't have to go in the back end and say, click, 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 add person X. That's just time that they don't have to spend. So turning that into a menu interface directly in the website has been actually amazingly popular. Very simple, based on AWS Cognito, not terribly complex. This also includes one of the big issues with cloud computing, which is cost anxiety. I want to make sure I don't get a $10,000 weekend. Allowing the PI to go in and set, this is how much money I'm willing to spend in a month, in a quarter, in a year. When I get to 50%, send me an email. When I get to 75%, send me an email. When I hit 100%, just turn off all the compute and make sure I don't spend way too much money. All the data stays there. We just turn off the compute. This actually happened earlier today on an account, which is good. And this is something that means you don't get surprised. And I think this also really helps with the communication. What am I getting? What resources do I have access to? Can I make sure that I'm not spending my own budget in a way that I'm not expecting to? So that's really important from the larger organizational sense. This, I think, is also a reflection of the way that our group is set up. So this is something that really makes the most sense when you think about a number of relatively autonomous groups operating with a fair degree of budgetary independence. We have over 200 labs. They each manage their own grants. They're each in charge of their own budgets. And so we really had to put the control in their hand, whereas if we were in a top-down organization, it might be, sort of insane to say, oh, the person at this level is able to say, I want to spend $10,000, whereas the approval might need to go up the chain. So this is also a reflection of what it's like to work at a biomedical research institution, which is essentially a collection of autonomous labs, which I think is sort of like a sociological reflection. The bit that I'm actually going to spend a minute on, because I have time. I have no idea what time it is. I think I have time. Okay, great. So I want to talk a little bit about interactive visualizations, and this is the last bit. So obviously, multi-QC is amazing. You run a workflow, you open up, you double click, and you can immediately get an idea of what happened. I think this is a great example of the type of visualization that makes the results more accessible, and they also just, it streamlines the process of doing science. Obviously, all the data is there, but when you have it formatted in a nice display, you can have someone say, I'm going to click run, or I'm going to just go log in to see the results that came off the sequencer. And instead of having to load something into Python or R, which maybe they can do, maybe they can't, instead of having to load something in and generate their own visualizations, instead of having to download data locally, the more that we can display directly in the web portal, the more value there is for the data that's being hosted there. And this is something that was, in a large way, one of the big carrots that we had for ourselves when we started building. This was actually one of the driving factors in the way that the entire system is built, is that we want to make sure that we can deliver data visualizations privately or publicly if you decide to publish the results, but within the context of collaborative research, that a defined group of people can access and view the data that they're generating. So I have some examples up here of different types of visualizations. So, multi-QC, so any sort of HTML, obviously. We put IGV into the browser, or we added IGV, so you can look at genome tracks, which is really nice. A molecular image viewer. At the bottom I have, bottom's IGV. Oh, right, in the middle we have a really nice charting interface. So taking tabular data, pulling it in, actually using DuckDB. So I really enjoyed that DuckDB talk. But being able to merge data and then put it into eCharts. So you can just go through a menu and say, I want a scatter plot. I want the X to be this. I want the Y to be this, and sort of set it up all yourself. I really like that sort of like flexible interface, and there's a lot more to do on that side of things. However, there is another part of it, which I'll show you right before I have my credits here, and this is an example of spatial transcriptomics. So, okay, this is something I'm really excited about. Spatial transcriptomics, you get a piece of tissue, put it in a microscope, take a picture, then you use a machine that measures gene expression across a bunch of different dots, so you can see what genes are being expressed in all these different spots. You do it with Space Ranger does the analysis, gives you a nice HTML summary, which is pretty good. You can actually download the data, a couple gigabytes, and load it up in the Loop browser, which is nice. So if you are, if you're comfortable moving that much data around, a lot of people use something called the Loop browser, which is cool. There's also this amazing library out there which lets you visualize that data directly in the browser. So this is an example of a public data set from the Cell Ranger website from 10x. But the idea is that you have all of this gene expression across lots of different spots. You also have microscopy data, and you can actually just zoom into it and explore it directly in the browser. One of the things that's really amazing here is that you can explore all the different genes that differentiate clusters of cells, like cell types. And you can mouse over them, and you can click on each one and say, I want to see where each gene is being expressed. And so you can imagine someone saying, hey, I did spatial transcriptomics. I'm really interested in this bit of tissue and this bit of tissue. So I'm going to go see what genes are expressed differently. I'm going to sort of I'm going to like leaf through them. I can color everything, make figures for your plot if you want to, and sort of like customize all this stuff. And maybe for some people, that's what they wanted to get out of this. Maybe that was the goal of doing spatial transcriptomics, was to really look at just identifying a couple of gene signatures and then moving on with your life. And if that were the goal, then they would be able to come here, look at this, explore their data set, and then move on with their life, having just done spatial transcriptomics done through our sequencing core without having to open the command line or download any files. So this is very early days. This is something that honestly is not actually in production yet. This is from our development side, so I'm not gonna say that we've solved this problem. But being able to complete that process of the data comes in, it's managed, it's analyzed, and then it gets put in front of a scientist within a secure, cost-controlled environment in a way that they can get what they need out of it to move on and not think about data analysis anymore. That's really my dream as a bioinformatician. And so that's something that I'm really happy to be able to have shared with you. We have a couple of examples of sort of what it's been like as we've scaled up. We've learned a lot of lessons as we've gone from a couple of users to a lot of users, from a couple data sets to thousands of data sets. I think I've covered most of these through the talk in general. And so to leave a couple minutes for questions, I want to say there is this much larger group of people that have contributed to this. Has been a massive team effort. I'm really, really thankful for the environment at the Hutch that we have, as well as the support we've had from AWS, which includes the recent AWS Imagine grant, which we're really lucky to have been recipients of this year. And I'll leave my contact information, and thank you all very much for your attention.
Thank you, Sam. That was a very impressive presentation. Very cool functionality as well. I have a question for you. Does Cirro implement any of the GA4GH standards for connecting to other repositories, data repositories, workflow repositories?
It's a very good question. So we have not yet. We are very interested in that. We've been looking at APIs to connect between different data sources. Actually, one of the first ones that we've been looking at has actually not been the GA4GH, but instead the FHIR API. So for connecting to clinical databases, which is something that's very interesting. But I'd say that something like GA4GH, and also in a larger sense, making this a data repository that is very easily accessible from the outside is very much in our plans for the larger architecture. So that's somewhere we definitely want to go.
That's wonderful to hear.
Yeah.
Very cool. Thank you so much. If there are any follow-up questions, well, we're going to have the coffee break now, so please give another hand of applause for Sam and the rest of our speakers this morning.